All right, I'm sitting out here outside the lab right now to discuss what our first dissection lab is going to be. And we're going to really take a look at the fungi kingdom and some mushrooms that I have procured from, that's right, Giant Eagle. So yeah, I got some portobello mushrooms from Giant Eagle, and we're going to take a look at these. We're going to take them apart. Look at this one. Ooh, look how big that mushroom is. We're going to take a look at these and examine their different parts. What makes them a mushroom? What parts do they have that put them in the classification in the kingdom of fungi? Usually when people talk about mushrooms, they are either a hard yes or a hard no with them. Do you want mushrooms on your pizza? People usually aren't, you know what, I'm not, it doesn't matter. It's usually a yes or no. Me, I love mushrooms. So we're going to get up close and personal with our friend the mushrooms. We're going to take them apart. We're going to look at their different parts and their functions. We're going to put them underneath the stereoscope and the microscopes. You're going to see a lot of the different tools of our lab. Now, why dissection? Why take a look at something so in-depth and closely? Why break it apart and look at it underneath the microscope? The biggest reason I can give you is because it's fun. It's fun to get up close with organisms and use the different equipment that we have in the lab. Another reason is it gives you first-hand experience with some organisms that you may not normally get up close and personal to. In another lab, we looked at protists. Those protists aren't something you normally get close to and involved with, but we did in that lab. Well, same thing with the fungi lab. Often you don't get up close and personal with the mushrooms growing in your yard or on the side of a tree or on your bread, but we are with this lab. And another one is I'm just curious. I'm curious to know what is going on in there. Look at that. It's kind of pretty. So I'd like to get up close and personal, and let's get into our lab, and I'm really excited to share it with you. So let me give you a quick overview of what's going to go on during this lab that we've got set up with our fungi here, our mushrooms. The main thing I want to do is I want you to get a good look inside of a mushroom, and let's use our equipment. I've got the stereoscopes and the microscopes set up so that we can look at these different parts, get a real in-depth, close look at the things that our eyes just can't see normally. And I'm going to take some pictures and show you those. I want to look at the different parts of what makes up a mushroom. All the different parts and names of the mushroom, what's inside, and kind of just an overall view of what fungi is. I'm going to be pulling apart these different pieces of the mushroom. We'll look at it under the stereoscope, we'll look at it under the microscope. And that's about it. I'll go over the equipment that I have here as I'm using it. So pay attention. I'll put any terms down underneath the video that you might need for the quiz that's going to follow this. So to get you familiar with the equipment that we're going to use, I've got our subjects, our organisms that we're going to be looking at today. I've got some forceps used to remove small pieces of the mushroom for today, or in other cases used to grab smaller items that might be difficult. I've got my scalpel. This is an extremely sharp, unbelievably sharp instrument used to cut things open, to examine them. Terrible story. My first year of using one of these, I was cutting an onion and caused myself to get about 7 stitches when I accidentally missed the onion and took my finger, took a piece of my finger. We've got some water and an eyedropper when we make our slides. Here are our slides that we're going to be using today when we make slides for the microscope, and these are cover slips that go on top of the item that we're going to take a look at. So the first thing I want to look at is these 3 mushrooms. They all have sort of different shapes, but very similar structures. This part right here, this is what we call the stalk. Up top is the cap. The other part that we're going to be looking at is inside, and those are called the gills. One of the things that you need to do first is to take your forceps, and there is some skin around this stalk that is called a veil, and that veil protects the gills. And when it's time for this to reproduce, that skin goes away, drops off, and then the spores inside will come out. So the first thing we're going to look at is this stalk. So I'm going to gently remove it. Ooh, break it off. There it is right there. I'm going to set this aside right now, and we're going to look at the stalk. The stalk is made up of small hair-like organs known as hyphae. Hyphae is basically what all fungus is made up of. Those small structures end up coming together and forming mycelium. So hyphae are these individual little hair-like structures. They're very tiny. And I'm going to pull some of these off, and we're going to take a look at them. So hyphae, very small hair-like structures that make up the stalk here and the entire mushroom. All these little tiny pieces together are called mycelium. So let's look at what hyphae looks like underneath the microscope. So in order to make a wet mount slide, we place what we want to look at on the slide. We take a drop of water on top. We take our cover slip, and then we just lay the cover slip down, and hopefully it pushes out the air, and we get a nice slide to look at. Before we take our slide over to the microscope, let's just examine what the mushroom looks like underneath our stereoscopes. Our stereoscopes are used for three-dimensional objects such as these, and for us to get a look at. So I'll take a look at the different pieces we have here, and you can see what I'm looking at. First thing I'm going to look at is just the overall entire mushroom. I'm going to look at the different pieces and parts. I will take some photographs of what I see through here, so you know what this looks like under different magnifications, and a lot closer than what your eyes can see. So the part I'm going to focus on right now is this stalk. The stalk is made up of individual little hyphae. Hyphae is the basic, it's what all fungi is made out of, and a bunch of hyphae together make up mycelium, which makes up the mushroom here. So we're going to focus on this stalk right now. Here is the stem of the mushroom. This stem is used to connect the mycelium down below the surface to the cap of the mushroom, which allows the mushroom to have its cap release the spores in order to reproduce. Our compound microscopes give us magnification of 40, 100, and 400 times. So I'm going to give you different shots of the hyphae underneath all three different magnification powers. This is part of the stalk at 40 times magnification. Here is a picture at 100 times magnification. And then finally we move up to 400 times magnification. And there you can start seeing the individual little hyphae that make up the stalk of the mushroom. Next part we're going to look at the cap of the fungus of the mushroom. This is the top part, the part that you first see when you see a mushroom out in the wild. We'll take a look at what it looks like underneath the stereoscope, and then we'll take a look at what it looks like underneath the microscope. Now, the problem with looking at things like this underneath the microscope is you need a very thin sample. So what I'm going to try and do is I'm going to try and pull off some of the tissue. That's right. These organisms have tissue. They're made up of all those different levels that we talked about before. There are cells inside this mushroom. Those cells make up the different tissues. We've got the tissue around the bottom called the veil, the tissue that covers the top of the cap. Those tissues working together make up the different parts, the cap, the stem, the gills inside. Those are all organs. And then finally we get to our organ system of all those parts working together to make the organism. So I'm going to peel off some of this tissue and I'm going to see if I can get a nice thin piece that is transparent enough to be seen through the microscope. Let's see if we can make this happen. Here I am at our stereoscope, and now we're going to go take a look at the cap of our mushroom. The cap of the mushroom acts as a protective shield for the gills that are inside the cap and to protect the spores that are held inside of those gills. So I think I got a piece of the tissue off the cap that is thin enough for the light from the scope to penetrate through and so that we can see it. Let me show you some pictures of what this looks like. This is cap tissue at 40 times magnification. Now we're gonna move up to 100 times magnification. And then finally, 400 times magnification. You can still see the individual little hyphae strands in that picture right there. The next part I wanna get to are the gills. Inside are these folded type of organ, and those gills contain all the spores that are inside of this mushroom. Those spores are what will help the mushroom reproduce. When it gets rid of those spores, it spreads them out around the area where the mushroom is growing, and hopefully some of those other mushrooms take root. Also, those spores are extremely light. So if it's windy enough, those spores, when they fall, will get caught on the wind and go. Because organisms don't want to compete for the resources around them. They don't want to compete for the food or the water that's around this mushroom. This mushroom wants its spores to get lost, go somewhere else in order to reproduce and grow somewhere else. So that way, a mushroom like this that could have possibly 40 million spores in it doesn't drop 40 million mushrooms underneath it, and hopefully, because those mushrooms would take over all the other resources around. So let's get into taking a look at these gills. All right, the next part we're going to look at are the gills underneath the stereoscope. Gills are a way for the mushroom to disperse its spores, and scientists use gills to identify different types of mushrooms. So I'm going to give it a shot of looking at the gills underneath the scope here, and hopefully I can get you some good pictures of what they look like at the different powers. So I was really excited to start looking at these. I looked at the same exact spot at 40 power and then at 100 power, and then here at 400 power you can actually see the individual spores sitting on those gills. And speaking of the spores, I've got a little bit of an activity I want to try with these spores that you could probably even do at home. You just need some common household paper and some mushrooms. So I saw this activity on the internet. And what we're going to do is create a spore print. We're going to cause the spores of this mushroom to come out and imprint and stain this paper that I have here. And I've got three little setups that I want to try. I've got a blank spot that I'm going to use. I've got two Post-its of two different colors, yellow and this nice salmon color. And then I've also got a glass slide on here because I want to see if I can get the spores to come out, and then we can look at the spores underneath the microscope. So here's what we're going to do. We're going to pull apart the stem of this mushroom, which allows us to see the gills underneath. And in those gills, that's where the spores are produced. These kind of mushrooms can have up to, they can have millions upon millions of spores inside that little area. So I'm going to pull this apart. I'm going to make sure that there's lots of room for the spores to fall out. So I cleaned up that mushroom pretty good. You can see I removed the veil from in front of those gills. And now I'm going to set this mushroom on my paper. I'm gonna give it a little bit of water right on top to cue it to maybe release those spores. And then I'm gonna cover it up so that the wind doesn't push those spores away. Okay, I'm gonna make 3 more just like that, and we're gonna let this sit for a little bit. All right, so there you have it. Let me give you a little close-up view of what I'm looking at. We've got 4 mushrooms, different types of media underneath it that we're gonna try and create these spore prints. Whoa, spinning all over the place, people. And we'll see what we come up with. Pinterest can't be wrong. It was on the internet, right? So we'll find out. So back to more fungi. So here I am after a weekend of letting these spores create prints on the paper. Let's take a look and see what we've got here. So if I pull up the one that's on the glass slide, yes, I definitely have a lot of spores, and we'll take a look at this underneath the scope in just a moment. On my paper, got some very dark spore printed paper there. A little bit darker than I wanted. Probably should have not kept it over the weekend. And here's another example of those spores that printed. And this paper right here, really covered. I'm gonna start removing some of these mushrooms and seeing how much spore activity we had. All right, this is an example of the paper that I ended up with. This one down here is really neat. That's a good one. And there's the rest of them. That's just a lot of spores that came out there. Millions upon millions of them. Let's take a look at what these spores look like underneath the scope. These are the spores underneath the stereoscope. The stereoscope magnifies up to about 30 times magnification. Kind of looks like a planet of some sort. These are the spores underneath the compound microscope. This is at 40 times magnification, and I'm going to try and zoom in on the same exact spots each time. Here we are at 100 times magnification, and the individual little spores are starting to be able to be seen. And finally, at 400 times magnification, you can see all the individual little spores there. There are just thousands upon thousands and millions of them on this slide. I hope you had some fun with some fungi. The fungus kingdom, we're going to wrap it up there. I hope you enjoyed that kingdom. We're going to move on now. You can take the quiz that follows this video.